Hey, we're back for Nervous System Basics lecture number 2. And we are starting with the structure of a neuron. This is a typical neuron. A neuron is more or less a cell with several processes. So I see my major portion of my cell body here, and I see a lot of sticky out things. The sticky out things are processes. Most of them are dendrites, which I see up here. And then there is the axon, which we talked about in lecture 1. We see him nicely insulated here by Schwann cells. All right, let's talk about these different parts. Starting with the cell body. The cell body is also known as the soma. Remember, soma, like we saw in somatic, means body. In the soma, I'm going to find the normal cellular stuff, like these beautiful mitochondria here. Plus, I'll find the 3 ends. I'll find the nucleus, the nissl body, and neurofibrils. We know what a nucleus is. The nucleus holds the chromosomes, the DNA. And the nissl body is actually just a word for the neuron's rough endoplasmic reticulum, which we know is very important for making proteins. And making proteins is going to be crucial to the function of our neuron. And there are actually structural proteins that are turned into these small little fibers that maintain the shape of the neuron. And those shape-maintaining fibers in the neuron are called neurofibrils. They're going to maintain the shape of our neuron. So these are some basic things about the soma, or cell body, to get us started with the structure of our neuron. Now, where are we going to find our somata? Soma is the singular, somata is the plural. Where are we going to find the somata? Most somata are found in the brain, in the cranial cavity. Second most are found in the good old spinal cord. The somata are where neurons do their processing of information. It is where they do their integration. If you recall that word from lecture number 1. And we keep the somata well protected, either in our skull or in our vertebral cavity. Okay. All right. Now, when I look at a brain, which I am doing right here, this is a frontal section of a brain right here. The brain will contain areas that are basically collections, groupings of somata, collections of cell bodies. Such a collection of neuron cell bodies, if it's in the CNS, it is known as a nucleus. Check it out, we got a brand new definition for the word nucleus. For example, this grayish cluster right here, I would consider that a nucleus, a collection of cell bodies in the CNS. Now I know what you're wondering. If I can have a collection of cell bodies in the CNS, can I have one in the PNS? And I absolutely most certainly can. Here's the spinal cord. If we're outside the spinal cord, we can see all these nerves here. And on these nerves, we have swellings like this one right here. There's another one right there, right there, right there. There's 4 there, another 4 over there. Each of those swellings is called a ganglion. And that is a collection of somata. In other words, neuron cell bodies, but in the peripheral nervous system. By the way, the plural for ganglion, if you want to say there are 8 in this picture, you would say there are 8 ganglia. Okay. Sweet. Let's talk about the processes, the things sticking out of the neuron cell body. First up, we got the dendrites. There are usually lots of dendrites, and I see so many dendrites here. Lots and lots of dendrites. Dendrites are the receptive part of our neuron. They're going to get signals, maybe from another neuron, or maybe from a sensory organ or cell. If you recall tactile cells from the integumentary system, tactile cells would signal the dendrites of a neuron. The word dendrites means branches, like branches of a tree, like this beautiful red oak tree here. And the purpose of branches is to do what? Whether it's in a tree or in a neuron, the purpose of branching is to increase your surface area. In my neuron, if I have more surface area, I can get more signals, I can get more information, and that is good. Now, the dendrites are going to send electrical signals. So like, let's say this dendrite was stimulated right here, okay? An electrical signal would travel down the dendrite to the cell body and onward to the axon. And the dendrites use a kind of electrical signal called the graded potential. We'll find them in the somata as well, but we find them in the dendrites too. Okay, so more on those electrical signals later. Don't worry. Let's talk about the axon. The axon is going to take those electrical signals that it gets from the dendrites and the soma. So the axon is going to get electrical signals, graded potentials, from the dendrites and from the soma. So graded potentials come this way. And graded potentials arrive at the beginning of the axon. The beginning of the axon is called the axon hillock. And it's this conical section right here. And then the axon goes from the axon hillock all the way to its ends. The ends are appropriately called axon terminals. And electrical signals go this way. The electrical signals used by the axon are known as action potentials. They are awesome electrical signals. So the action potentials will go from the axon hillock all the way to the axon terminals. Now, there's almost always just a single axon per neuron, whereas there are many dendrites. So it's like, you can think about dendrites as the input for a neuron and axons as the output. Action potentials, electrical signals that are awesome, go down the axon all the way to the tips. The tips are called the axon terminals, and they are going to release chemicals called neurotransmitters, maybe onto another neuron where they can stimulate graded potentials in it, or maybe onto an effector cell like a muscle cell where they could stimulate it to contract. Now, a few more things. If we look at this axon here, sometimes the axons have big branches. This guy does not have any big branches, but if he did, a big branch would be called an axon collateral. We do see the insulation. These blue cells, each of them is a Schwann cell. We met that kind of glial cell in lecture number 1. The outside of a Schwann cell is called the neurolemma. This is a good vocabulary day, isn't it? Who am I kidding? Every day. Every day is a good vocabulary day. All right, there are actually gaps between the Schwann cells. We can see these gaps right in here. Dun, dun, dun. Lots of gaps. The gaps are called nodes of Ranvier. Nodes of Ranvier. And they're going to be crucial to how action potentials, those electrical signals, travel down the axon. Now, at the end of the axon, we do get some branches. Branches at the end are called telodendria, which literally means branches at the end. And then the telodendria finish as axon terminals. And as we said earlier, axon terminals release neurotransmitters either onto another neuron or onto an effector cell. All right. Let's clear that. Let's keep on going. Now here I see some neurons meeting. Where a pair of neurons meet, that is called a synapse. So it's a junction between two neurons or between a neuron and an effector cell. And I see that here. Check out this neuron that I'm shaking my cursor over right now. I'm on the cell body, the soma. Over here I have another neuron's axon. I see his Schwann cells. I see its telodendria and its axon terminals. Where these axon terminals meet the soma, each of those junctions is called a synapse. So I have an axon synapsing with the soma. Over here I have axon synapsing with the dendrites. Down here I have an axon synapsing with another axon. This is where neurotransmitters are released and we're communicating between neurons or between neurons and an effector cell. If you are moving, you are using synapses. If you are doing anything, you are using synapses. If you are studying and learning, you are literally making new synapses. If you fail to use those synapses again, they will wither away and die. This is why periodic studying is good. This is why cramming is bad. You gotta review material, space review, study, and then repeat it later, and then repeat it later. Anyway, make new synapses, don't let them die. You know what else helps synapses not die is getting a good amount of sleep and not being too stressed. It's good advice I'm giving you here. Get some sleep. Don't be stressed. Really easier said than done. All right, my friends. Let's keep going. Zooming in on a synapse right here. What do I see? I see the axon of one neuron. We're calling this guy the presynaptic neuron because he's taking information to my synapse in this case. I got my axon terminal down here. The axon terminal is going to have little membrane bags called vesicles. That should sound familiar. The vesicles are filled with these green neurotransmitters. When an action potential gets here, it prompts the synaptic vesicles to undergo. I'm going to start spelling the word. You're going to shout it out. Did you say it? Exocytosis. And that releases the neurotransmitters into this gap between the neurons, and then they diffuse across, and they're binding to these integral proteins here. Remember those? They're also channel proteins. The neurotransmitters are making them open up, causing the cell body, this is the soma here in my postsynaptic neuron, to experience an electrical signal. This guy's the postsynaptic neuron because he's getting the communication in this synapse. So this is one single solitary synapse here. You are going to have trillions of synapses in your body. Okay, one more little piece of anatomy. This gap between the two neurons here, it is known as the synaptic cleft. The synaptic cleft, because it is just a small little space, the synaptic cleft. All right. We'll revisit this later as we talk about action potentials and what they do. So don't worry. Okay. What we're going to do now is classify some neurons in terms of the kind of structure that they have. And there are three basic structural types of neurons: multipolar, bipolar, and unipolar. I can see the cell body right here, cell body right here, cell body right here. Can you just figure out already, without me even telling you, which is which? I bet you can. I bet you're doing it right now. In fact, what do you think this guy is right here, this dude? What do you think he is? You're right. He is the unipolar neuron. He's called unipolar because his cell body only has one process coming out. It quickly branches, that's true, but one comes out. All right, what about this fella right here, this guy? Ta-da, that's a horrible looking arrow. This guy right here. Bipolar. He's a bipolar neuron. He's got two processes coming out, and they both branch later on. And the last one, by process of elimination, must be the multipolar one. Lots of dendrites and an axon. So these are the three basic types I'm going to see. Multipolar, bipolar, and unipolar. The most abundant one, the one you have so many of, is the multipolar. Second most abundant, so number one. Second most abundant, number two is the unipolar. Number three is the bipolar. I probably should have put them in that order. I went like many, two, one, many, two, one. But in terms of abundance, just remember that good old word mub. Excellent vocabulary word mub. All right, let's talk about the different kinds of different structural kinds of neurons. Let's do that for a bit. So, multipolar neurons. Almost all of your neurons are multipolar neurons. I mean, seriously. I really think the number is like 99%. I'd throw some cash that it's 99% of your neurons are multipolar. If we're talking motor neurons are all multipolar neurons. There are these neurons in your brain where you do a processing, there we go. They're called association neurons. They're all multipolar. The only thing that's generally not multipolar neurons are sensory neurons. So, multipolar neurons are going to have a single solitary axon, dun, 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 there it is, and many dendrites. This is a multipolar neuron here. This is his cell body right here. This is probably his axon down here. I can't say for sure, and these are probably dendrites. But one axon, many dendrites, that's a multipolar neuron. Most of the times when we look at neurons, we're going to look at multipolar neurons. Like if you Google search neuron for an image, you'll probably get a multipolar one. And that makes sense because, again, 99% are motor neurons or association neurons. Those are multipolar. Okay. Bipolar neurons. Bipolar neurons have a cell body, a soma. Then they have a single dendrite, which itself branches quite a bit, and a single axon, which branches at its end. So axon's pretty normal. The dendrite is weird because all the branches are going to come together and then come on down to the cell body. We were used to seeing whoops, I didn't want to do that. I want to go back. There we go. Lots of dendrites all coming into the cell body. The bipolar neuron does not show that, does not do that. All right, our bipolar neurons are going to be special types of sensory neurons. And we're going to find them in two places. We find bipolar neurons in the back of our eyeball, in our retina. We're going to find bipolar neurons. You can see these guys right here are in fact called bipolar cells. They are bipolar neurons. So we find them in our retina. We are also going to find them in our olfactory epithelium. Let me write that down. Our olfactory epithelium. Remember, the word olfactory has to do with smelling. And the olfactory epithelium is in the superior nasal cavity. We are looking at olfactory epithelium as we zoom in on this box right here. So this is our olfactory epithelium right here. And you can see these yellowish guys. These are bipolar cells, bipolar neurons. So they're involved in vision in our retina and involved in our sense of smell in our nasal cavity. And of course, what bone do we have right here? What bone is this? Right there. What bone is that? I'm going to label that bone A. And I'm going to label this bone right here. See that guy? Right there. I'm going to label him B. Email me. Tell me what they are. Each is worth 1 extra credit point. Oh my gosh. That's amazing. A is easy. B is hard. All right. Good stuff. Good stuff. Bipolar is done. Let's do a unipolar neuron now. It's got the cell body. It's got a short process coming out of it, then splits to go 2 ways. And this entire thing here is the axon. The axon starts right here and goes all the way right here. So what's weird about this guy is the dendrites fuse together. So here are the dendrites. The dendrites fuse together to make the axon. Then the axon goes down, and passes the cell body along the way. So in this case, the cell body is not part of the electrical pathway. The signal would go from dendrites, all the way to the end of the axon. In both of the other neurons, multi and bipolar, signals would go dendrites, cell body, axon in that order. Not in this guy. We skip the cell body, so to speak. Now, the axon is divided into 2 parts. The part that is going to be between the dendrites and the cell body, that's called the peripheral process. And then the part between the cell body and the axon terminals, that's called the central process. Which part do you think is closer to the spinal cord? Did you guess it? The answer is the central process. The central process is actually going to go into the spinal cord, which of course is part of our central nervous system. All right. So, where do I see these guys? All the sensory neurons going into my spinal cord are going to be unipolar neurons. All the sensory neurons that are taking sensations like touch, pressure, pain, temperature, proprioceptive signals as well, all the sensory neurons going into my spinal cord are going to be unipolar. So this guy right here, this is a unipolar neuron. He is labeled as sensory neuron right here. Here are his dendrites, which fuse to make the peripheral process of his axon, taking information in, that's why we say afferent, past the cell body, onto the central process, on into the spinal cord itself. So my sensory neurons that are going into my spinal cord, so any sensory neuron from my body going to my spinal cord is going to be a unipolar neuron. And now notice that in this picture there is just a single solitary cell body right here. Of course, there would be hundreds and hundreds and hundreds. So this region right here, which is kind of wider than the surroundings, is going to be a cluster of cell bodies. And that, of course, is called a ganglion. We met that term in lecture number 1. No, we met it in lecture number 2, didn't we? Yeah. Either way, we met that term before. All right, we've done our 3 basic types. Then there's the functional classification. We're going to have this functional classification based on the 3 functions, which you know already. The 3 functions were sensory input. So 3 functions, right? Sensory input, integration, and motor output. Sensory neurons are going to do sensory input. That is a functional class of neuron. And notice that this sensory neuron depicted right here, this blue guy, is a unipolar neuron. One process coming out of that cell body. Then, Integration. Well, this neuron that's going to do the integration, he is called an interneuron. We will also see neurons like this labeled as association neurons. So the second functional type of neurons I'm going to have is known as an association neuron, also known as an interneuron. I like the word interneuron because notice he is between the sensory neuron and our third guy here, the motor neuron. The motor neuron is, of course, responsible for motor output. Now, both of these guys, my interneuron and my motor neuron, there we go, they are both multipolars. They are both multipolars. All right, we've now done the structural and the functional classifications of our neurons. And guys, you know what we are ready to do? We are ready to figure out how neurons are going to send electrical signals. We know neurons are going to send electrical signals. An electrical signal will go from one end of a neuron, so like from this dendrite tip right here, all the way to this axon terminal right here. An electrical signal will run all the way like that. Okay? And now in order for neurons to use electrical signals, neurons are going to have what is called a voltage. Neurons have to have voltage. You know this word, of course you do. So neurons have a voltage. They have this electrical difference between the inside and the outside of the cell. The voltage of a neuron is known as its membrane potential. Because obviously, if it's a difference between the inside and outside of the cell, what separates them but the membrane? The word potential here applies to the fact that we have a source of potential energy. Okay. Now, we can actually measure this voltage. We can take a voltmeter, which is just a device that measures this electrical difference, and we stick this very tiny electrode inside a cell called a microelectrode. We can measure the voltage of a neuron. I've actually done this. It is a very tedious, unfun job to do. I don't recommend that you do it. I did not enjoy it. Anyhow, if we did this, we would get a reading of the electrical activity inside. In this case, we're on the axon of our neuron here. It's our axon here. And we can see there is this electrical situation, this voltage, and has a value of negative 70 millivolts. So the inside of our neuron has an electrical state that's negative 70 millivolts different from the outside. Okay, great. What does all this mean, though? Well, this neuron right here that I'm dealing with, this neuron right here, it's just chilling. This neuron is at rest. So if a neuron is resting, it is going to have this resting value called a resting membrane potential. And the typical value for that is negative 70 millivolts. That is not a lot of electricity. Okay? Go get a 9-volt battery from somewhere in your house. You got one in your smoke detector. Just make sure you put it back. If you were to lick that 9-volt battery, if, this is hypothetical here, you are not going to do this and tell me you got hurt and blame me, okay? If you were going to do that, you might feel like a little tingling on your tongue. And 9 volts is 9,000 millivolts. It's not a lot. It's negative 70 millivolts is not a lot, okay? But it's enough, and it matters. Here's the bottom line, okay? Neurons, when they're at rest, they have this electrical state called a resting membrane potential. It has a value of negative 70 millivolts. Okay, keep that in your brain for a second. Keep that somewhere in your brain. All right. What we got to figure out is two things. Number one, where this resting membrane potential comes from, like why it exists, like how it's created, and then also how we utilize it to send electrical signals. Okay? And that is what we are going to do in the next lecture. I'm going to quit right here. I like this little half-hour idea here. I'm going to quit and pick it up next time. So I'll see you guys later. Bye-bye.